So it's sort of a CASP talk, but I want to do it a little bit differently. In the CASP conference that we have at the end of every experiment, we ask people who've done reasonably well, got interesting stuff, whatever, to give a talk, but we give them strict instructions. You should focus on what went right, what went wrong, and what you learned. And so I'm going to try and do that for CASP itself, with the purpose of seeing where things can go next. I mean, I think that's the spirit of this session. How is science going to evolve in our area as new technologies become available? And that's essentially it. If I press this, it changes. So I'm going to talk about the problem which I think everybody in this room knows. In principle, you can calculate the three-dimensional structure of a protein from its amino acid sequence. And this has been recognized this is from Amundsen in 72, but it was sort of recognized before that. So it took about 50 years to get to the AlphaFold solution, which I expect everybody knows about. I want to talk a little about what happened in those 50 years. So first of all, as everybody I think appreciates, it's a hard problem. It's got a very large search space. You can pick any 10 to the whatever number you like. If you're trying to do it in real space, it's a very frustrated landscape. And it's a very subtle free energy difference between the folded and unfolded states. So you need, in some sense, a very accurate potential. I'll come back to the significance of that later. And a fourth point is that it's hard because it's hard to get the science right because you're working in a virtual world you can manipulate. And you'll see what I mean by that in a minute. So 50 years, a lot of methods. This is an approximate tree. This thing, yeah. So methods based on structure principles, things we thought we understood, search methods of different sorts, physics, of course. I'm an ex-physicist, so I really like this branch. Pity it didn't work. And these are evolutionary relationships, which, as we all know, I think, came to the where we got to deep learning. I don't want to go through them in detail. I want to illustrate a couple of things. First of all, a lot of this is really good basic science. You have an idea. I think genetic algorithms are going to be useful. You test it. It takes quite a long time. You've got the idea. You need to develop an algorithm. You need to develop an engineering of the software. You need to see if it works. So long turnaround times are almost inevitable, even when you're doing good science. So let me focus on that genetic algorithms thing. The first paper on that for proteins was published in 1993. And in CASP, we're looking at all this stuff, as I'll get to. We saw that a version of that, the fragment assembly, which is just above that in the structure thing, actually worked to some degree. So basic idea, new search method, a lot of development applying it, and then in 2002 or so, you see this sort of result. So on the left here, you have a library of protein fragments. So these are small bits of proteins which you see frequently in structures. And on the right, you see a predicted structure in green on the blue experimental version. Very good agreement. This is actually from the Baker lab by putting this stuff together. Look, really, really promising. And again, the science is the science. It actually stalls at something like 80 to 100 residues. Wonderful for getting main chain, fold right, then can't scale. Don't know why, but that was the end of that story. It was really a nice, satisfying story. Let me talk about another one, and that's the very top thing there, pathways. So this was something which partly is down to somebody we already heard about, Cyrus Leventhal, who is, I want to emphasize, I really agree with what's been said about his pioneering work in using computer graphics for this field. I think it was really influential and drove things forward. I'm going to say something slightly less flattering. So Cyrus published this paper in 1968 in which he said, because the search space is so big, there must be, the text up here, a sequence of events that follow one after another to carry out the protein from where it starts to the folded state. And I and many other people were enamored with this, both experimentalists and computational people. In the next 20 years, I spent quite a lot of time on this idea. It's wrong. I mean, basically, it assumes the landscape looks something like this. You don't know where you're going until you drop this. Probably can't see this. This is where the hole is for this putting green on a golf course. You don't know where you're going until you get there. And of course, that's not true. As you go places, you go down in energy. It's not following a pathway. It's a statistical process. And I think today, very few people would buy into what Cyrus said. My point in telling this negative story, and there are going to be a couple more, is that why was that? Why did we spend so much time on something which in the end really didn't pan out? Come back to it. Okay, let's go on. So things moved along, and it looked like we were making very good progress if you looked at the literature. So these are two papers when we're sort of 20 or so years into solving the problem, and you can see actually it's solved. Here we've got 2 angstroms RMS. Here we've got something that can do all sequences in the human genome. Actually, as we know, this happened in really 2020 and 2021 with AlphaFold 2. This is a paper in Science, so it's not some sort of out-of-the-way journal. And you can see I was doing this along with my colleagues. So I pointed out what the difficulty is here already. The peer-reviewed publication system didn't, and I'm going to explain, doesn't work in this area. And again, I think, spend a long time on it, it's to do with the virtual world in which you're doing the calculations. But this is the relevant fact. It didn't work. We didn't know where we were. We didn't know where we were going. So that's the origin of CASP, which was started in 1994 by myself and Christoph, who is here. These are current organizers. Andre, if anybody's involved in CASP will know he's the guy. Anna, for a long time, really drove the organization. She was the adult in the room. And then other current organizers, Torsten, who might not be here, and Maya. The idea of CASP was, can we get more rigor than we were getting from the peer-reviewed publication system? We'd like to do that transparently if possible. And the bottom line is we want to be able to trust what we say about what the methods can deliver. Very fancy, but the idea, of course, is very simple. You look at what experimental structures are about to come out. You ask people to give you models of those. You compare the two, and you've got very strong evidence, one way or another, about how a model performs. There's a lot of stuff in CASP, a lot of people involved, but one set of people are really important, and I want to make sure to mention them, and that's the people who make sense of this. We refer to these as assessors. They're not part of the organization. They decide what the results mean. And to me, this is a very important part of why CASP works to the degree it does. So, experiment every 2 years, typically about 100 targets, about 100 research groups taking part. So we get, it's not really statistically significant, but we've got a very good measure of what's working and what isn't. And I want to switch now to the story of what did work in the end. Of course, that's the deep learning. But the deep learning very much comes from earlier work on contact prediction. So, and that's another cautionary tale, which some of you have heard before, but I want to tell it again because I think it's important. So we usually think about a protein as a set of 3-dimensional coordinates. Actually, if you define the contacts within the protein, you've got another really full representation of what's going on. And these things have been around since I was a graduate student, known as contact maps, originally referred to as OI plots because he was the person who came up with them. So sequence by sequence, filling in matrix elements like this one here, roughly 160 to either what it is, 20, where things are in contact. If you can somehow derive these, it's a relatively simple calculation to get some form of the 3-dimensional structure. And how do you derive them? This largely goes back to a paper by Chris Sander, Alfonso Valencia, and others in 1994. It's not actually their picture, but it makes the points they made. I want to know what a structure is. There are contacts in it. If there's a contact and I've got a sequence alignment of the family, there are going to be constraints on the pairing of the amino acids I can put here. So here's an example. You start off with a big positive with a small negative. You change as evolution goes across the phylogenetic tree to a slightly smaller positive and a slightly bigger negative. Here you switch to 2 hydrophobes of about the same volume. So you're constrained about what you can put there. And the idea in this paper was that you look at all columns in this alignment and simply calculate with something like mutual information whether or not there's a contact there. This was 94. We just started CASP, too late to put it in from the beginning. But as we've done with a number of areas, we created a special category for this starting in 1996. And so here's the record of the precision performance of those methods. This is 1996, this is 2016. So started off slow, precision went to about 20%, which is pretty bad, and stayed roughly the same until a big jump in 2016. What was going on here? Why were we so long in this state before we got a big jump? And the answer mostly is that we were doing the statistics wrong. That thing I told you about correlated columns is a flawed approach to the problem. And it's a flawed approach because there are knock-on correlations from things that aren't in contact. This is from the first time I was aware of this in 2010. Here's a couple of amino acids, I and J, not in contact, but there's a chain of contacts linking them. And so you get this residual correlation. And in fact, typically there are many such chains linking an I and J, and so they build up to be bigger than the actual driving contact ones. So this actually is an old problem in statistics. It was first published in the area of protein structure in 1999, so before most of this. Why didn't we realize it? Why didn't people start using the right statistics? And because it's an old problem, once you realize you could do something, you should be able to get somewhere. I think the problem was that there were a number of ideas floating around about what was actually going on. I count 5 of them. The first one is the idea, well, you've got a lot of mistakes because it's not true that all of the proteins in a family have exactly the same structure. Some have different contacts. So you're not going to get a good result. I actually believed this. It's nonsense. What the problem we were getting was false positives. What this would give you is false negatives. Then some people said, well, we've got to do these pairwise things better. Other people said, gosh, it's a phylogenetic tree. You idiots are treating all these sequences equally. That's the problem here. We've got to treat it properly. Some people said, well, there's correlations with function. There's all these long-range effects, and they're all wonderful. And then there was the right answer. And in those years, we were meeting at CASP. We were talking about this stuff. There was no occasion in which we sat down and said, okay, let's think about what might be wrong here. What are the different ideas? What calculations could we do to test each of these ideas? And actually, if you look at it that way, it would have been pretty simple. We never did it. Why didn't we do it? I don't know, but we've got to change in that sort of way. Okay, so much for the second negative. Let's go back to the positive. And as you all know, this stuff with contacts led to progress. So here's where we just were. This is the modified contact methods, doubled the precision, but still not very good. And then there was the realization, well, realization was there probably before, but the realization of the fact that this is a two-dimensional image, and two-dimensional images were doing very well in another neck of the woods, deep learning using convolutional neural networks. This is an example, training a network to tell the difference between cats and dogs. You put cats and dog pictures here, you put yes or no here. The equivalent is you take all of the experimental contact maps, put them here, you put the multiple sequencer line here, you train the thing, and you predict contact maps for new structures. As we'll see in a second, that took us to getting most of the folds right for the CASP targets. It was a major step forward in 2018, but not the detail. As with all of the other methods, the detail wasn't there. And everybody's aware, 2 years later in 2020, DeepMind came back with AlphaFold 2. I go into the technicalities here, but essentially the important thing here is attention. Which elements of the data should you be paying attention to? Whereas that was done with these convolutional effects in the old networks. Here, you actually learn where the important relationships are in the data. One point to note is that this actually outputs not a contact map, but a probability of the distances between particular amino acids. This is a relevant point to where we'll get to if I have time. How am I doing for time? 15 minutes. We might make it. Let's see. Okay, so this is an AlphaFold slide looking at a target. Here's the residue. here's what comes out as actually AlphaFold 1 of the predicted distribution of distances between that residue and a number of other residues. So for some things, you get a very peak distribution, so it's essentially predicting a contact. But in other places, you get this distributed distribution. It's still got a lot of information, but you don't know exactly what the distance might be. Now, for those of you who are familiar with this sort of stuff, that's very reminiscent of potentials of mean force, where essentially you can, instead of believing in, let's say, a physical charm force field, I want a force field for a protein, I'll just look at what the observed distances are between particular atom types. And I'll use those distributions, because of the Boltzmann relationship, they're equivalent to free energies. I'll use those distributions as my force field. So you'll see in a minute why I want to emphasize this. But before we get to that, let's look at the overall effect of these different methods across the CAST. This is a simplified view of how each of the things I've mentioned built up to where we've got to with single proteins right now. So this is how much you could use homology modeling, the sort of thing Barry was talking about. This is agreement of main chain. This is a 90 agreement on this scale between experiment and calculated structure is essentially as high as you can expect to get. So solving is having a line at 90. And this is the AlphaFold 2 in 2020. It's almost there. We could discuss the details, but it really is a solution. I'll show you a little more in a minute. But we started down here in 1994, and 20 on this scale is essentially random. So we went from random to a solution in 16 years. Along the way, this is the effect of the fragment assembly mostly. This is the effect of the improved contact predictions. This is the effect of the convolutional neural networks. Let me sum up this in pictorially, and then I'll make a point about all this in terms of this what went wrong thing. So just to summarize that, here we were, this is random predictions in 1994, another version of the fragment assembly. This is a predicted and experimental structure for the improved contact predictions. And then these are two examples of AlphaFold. And so a very good main chain agreement, but the really extraordinary thing is that very often you have this atomic level agreement. This is going to be important in something I'm going to say in a minute. So isn't that wonderful? Yes, it is. We have a solution. But why is it that DeepMind got the solution? Certainly, they're very smart and able. Don't want to in any way diminish that. But some people have said, Google, you know, they've got money coming out of their ears. Of course they could do things we couldn't. If you looked at the number of groups also in CASP who were trying to do this and you added up their resources, I don't know this for sure, but almost certainly, cumulatively, we had more resources than DeepMind did. The difference was DeepMind was sitting around a table once a day or a week or whatever they did and intensively discussing their different approaches and how they compared. And again, as a community, we didn't do this. So coming back to that in a minute. So I'm going to run out, but let's try. I do want to say a bit about what's going on here. Now, I think we don't really know what's going on in terms of how these methods work, but I think we can see some things. And let's try and look at something. Okay. So why do deep learning methods work? I came into this with the contact story, with the alignment story. So perhaps it's the alignment. And some people in the heated discussions about this say, of course it's the alignment. These methods depend on having a sequence alignment. Actually, that's not true. For about a third of the cases, a single sequence works very well with AlphaFold 2. So it can work without the sequence alignment. It's not reliable, but from a sort of principle point of view, it can. Other people say, well, it's, you know, it's sort of an update of fragment assembly. It's just cleverer fragment assembly. How can you address that question? One way is to say, well, if it's fragment assembly, I'd expect things to go wrong when there's unusual features in proteins. So my colleague Osnat and wife, Osnat Hertzberg, looked at this, went to the 2020 experimental structures and asked, what are the rare features here and how did AlphaFold 2 do? This is an example. I don't have time to go into the specifics, but you can see it's a kinked helix because of a pi turn at this point. There's no proline here, which is typically what you see. And yet the machine got it very right, and it's very unusual. Now, what went on here, this is a strain, but if you put in this kink, you rearrange the side chains at this point into what looks like a much more energetically favorable arrangement. So the machine has somehow spotted that by doing something unusual, you can compensate and get energy back somewhere else. That's not the sort of thing you can do with fragment assembly. The other extraordinary thing here is the accuracy. How can you get atomic accuracy? It's much, much better than we saw with any sort of force field, any kind of molecular dynamics refinement. And the answer I suggest, which will get me into trouble, I'm sure, is that effectively the machine learns a potential of mean force between every atom type in proteins. And it's a very good potential of mean force because there's lots of data, lots of parameters, many more than in CHARM. Here you've got millions of parameters. CHARM has, I don't know what it is, 100, let's say. It's a much more nuanced representation of the energy, and so it works. Anyway, I should get on because I do want to finish up with the punchline. So that was 2020. 2022, another CAS, we said what things now, which weren't accessible, might be accessible with these sorts of methods. And we looked at 5 of them. We still kept proteins. This is complexes, which we've already been looking at with our sister organization, Capri. This is multiple conformations. This is an RNA, but most of the data were proteins. And I want to say a little bit about how these worked. But we did 2 other things. This is protein-ligand interactions. And then at that point already, there have been a number of papers on this. In fact, it turned out that the classical sort of ligand docking methods were quite much superior to the deep learning methods that have been put out at that point. This is RNA structure. And the same thing, when we looked at this in the CASP results, the traditional methods were better, and they're not all that good actually, are better than the new AI methods. So the thing I told you about in 1990 is still there. As a field, we are still publishing things which are misleading. They're not valueless, but they're misleading. Okay, so let's just look quickly at the sort of success stories here. This is, by the way, this is from SG, who was the assessor here. So these are her slides. 3 examples, one for an intertwined dimer, one for this big hairy beast, and one for a nanobody protein interaction, all very well represented. Tremendous progress compared to what we'd seen in CASP before. And SG captured this with this plot. It's like the plot I showed you before. This is how much you can use homology. This is a measure of how accurate the contacts are, both precision and recall. And you can see here's where we were in 2020 before people really used these methods for complexes. Here's where we were in 2022. It's not a horizontal line, but it's gone sort of halfway there. So we're very interested in what will happen next. Quick on multiple conformations, because this is a very hot topic. Remember, this is 2 years out of date, but still what we saw is that this is an ABC transporter with 3 conformations. These are 2 of the experimental ones. It gets too clumsy to show you the whole thing. And people were able to get these 3 different conformations with different ligands bound. You might not be impressed by that because there are similar structures of ABC transporters, not the same one, in the PDB. So maybe it just picked them out. But this is, I think, more impressive. This is a simple homodimer enzyme. You come down the C terminus here, you go into this bronze tail, which interacts with the opposite subunit and finishes up interacting with itself. For other reasons, the people working on this introduced a mutation here to destabilize that part of the thing. And what happened was this C-terminal tail swung around and went over here and followed what was before the path of the other subunit. This is not in the PDB. It's completely new. 5, 5, 5. All right, we're going to make it. Well, might be any questions. Yeah, not in the PDB, and a number of groups were able to do this. So still a very hot topic. We're going to see in a few months how this CASP does, but very encouraging. Okay, so that's the good news. I'm going back now to the bad news. I've told you 3 stories of things that, in my view, went very suboptimally over the years. What could we be doing about that? I think there are 2 sorts of things. One is, can we do something to do better at being able to criticize papers in a constructive way? Not to be destructive, but to do better than peer review does. Why would you want to do that? There are hundreds of papers coming out now on deep learning. I, for a time, was getting notified of one a day, and I'm sure other people were as well. It's gone down to about one every 2 or 3 days. It's totally overwhelming. I have no idea, lots of interesting stuff, but I've no idea how to take it. What would you like to get out of that? You'd like to be able to know, for example, this is showing you sort of core technologies. Of course, there are a lot more than this. This is showing you potential applications. I happen to be very interested in mutant effects. There are all sorts of other nuances to this. How could we know, is this a good method for what I'm trying to do? Is this paper really helping me, or what's going on? So what I'm proposing is that we should set up, first of all, a community effort to look at papers in a structured way and in some sense score them. This is going to be controversial, I know. Here's an example. I'm using the diff.dot paper. Some of you may know this from last year. And let me emphasize, I am not attacking this paper. I think it's actually quite a great paper, but it's not perfect. So there's a series of particular queries, and each query gets scored. So for example, the ideas here are really great, gets a 5 out of 5. On the other hand, the evidence that it really is an improvement in terms of performance is actually weak, in my view. So it gets a 0. So great paper, very worth having, but you should not believe, in my view, the results. The idea is that we could actually keep a record of this publicly. There'll be a lot of arguing and comments, that's okay, to be able to know where we're getting to. And the other thing about this, I have no data on this application, I'm doing it in a different area, is that if you put up this kind of structured stuff to look at something, you can actually devise prompts for the current AI tools, which are not perfect, but will do a lot of this for you. And so the problem of scale here, he's looking at me, is he? He's looking worried. The problem of scale here can be solved. So, final slide. We used to do science a particular way. It had lots of problems. We've gone on a lot. There are a number of new things here, but I don't have time to go through it. There are a lot of things we're now moving into, and where we should be looking at what will work and won't work, and thank you.
We may have time for one question.
Spoil sport. Well, that's easy.
John, thank you very much for the very interesting talk. So as you mentioned that AlphaFold largely solved the monomer structure prediction problem. So CASP also has to be changed. What are the new directions of the CASP?
You were asleep. I showed a slide of that.
Yeah. I'm talking about the CASP 16. 16.
Yeah. So it's the same set, right, as you know. We basically tried to move on to, I said, the areas that might be promising. So that's ligands, RNA structures, complexes, proteins and complexes with RNA, and the multiple conformation stuff. And I think a lot of papers, as you know, I think it's going to be very interesting to see in a couple of months or 3 months exactly how things have worked out.
Thank you, John.